Okay, it's 3 o'clock Eastern Time, so we're going to go ahead and get started with the webinar. Welcome, everyone, and thanks so much for joining us today. My name is Willie Goldsmith, and I'm a Senior Associate with the Lenfest Ocean Program. And for those of you who don't know us, the Lenfest Ocean Program is a grant-making program that funds research projects and expert working groups to address priority science needs that face ocean and coastal decision makers. And if you'd like to learn more, you can visit us at lenfestocean.org, where you can also sign up for our newsletter. And please also follow us on Twitter. We are at Lenfest Ocean. For today's webinar, we're excited to have with us Bryan DeAngelis of the Nature Conservancy, Philine Ermgassen of the University of Edinburgh, and Jonathan Grabowski of Northeastern University. And today they'll be introducing their new project looking to better estimate how oyster reef, seagrass bed, and salt marsh habitats enhance fish and invertebrate production. And as part of this project, they'll also be looking into which environmental factors, as well as which biological characteristics of those habitats affect their production. Today's webinar is what we at Lenfest like to call a launch webinar. And that is a webinar as the work is just getting going, just starting, just to announce the project and introduce it while also engaging from the beginning managers, policymakers, scientists, and stakeholders. Ultimately, this work could help managers better target their habitat conservation and restoration measures that both maximize the benefits of ecosystems and the economies that rely on these coastal habitats. So before I turn it over to Bryan, Philine, and John, I just wanted to go over a few quick logistical items. First off, we have all attendees on the line are muted. This is just because we have so many folks who are on the line and we want to prevent any feedback or echoes from occurring. And after the presentation, we'll have about 20 or 25 minutes for questions. And if you do have any questions, you can use the Q&A panel on WebEx to type and submit your question at any point during the webinar. I'll keep track of the queue and read the questions aloud to the researchers at the end in the order in which they were received. And depending on how many questions we do have, we might not get to all of them, but folks are, of course, welcome to follow up with us here at Lenfest or with the researchers themselves, and we encourage you please to reach out. For next steps, we are recording today's webinar, and in the next week or so, we will be making the webinar recording with the video of the PowerPoint available online on our website, and I'll be distributing that link via email. I do have a running distribution list for information, for distributing information about this project, and if you're not on that, or if you're not sure if you're on that, please feel free to just shoot me an email. My email address is on the slide you see right now, and we'll get you on that list. We'll also be live tweeting today's webinar using hashtag DeAngelisWebinar, and feel free to use that as well if you're inclined to engage with us there. So I'm now going to turn it over to Bryan DeAngelis. Bryan, I am transferring presenter privileges to you now. All right, that was great, Bryan.
All right, wonderful. Okay, well, thank you everyone for being here. We've been working on this project for a number of years now, and so we're really excited to be launching this new phase of the project with support from the Lenfest Ocean Program. So this is our team of principal investigators. My name is Bryan DeAngelis. I'm with the Nature Conservancy's North America Oceans and Coast Program. Jonathan Grabowski with Northeastern University and Philine Ermgassen with the University of Edinburgh are my co-PIs. And we want to acknowledge a fantastic research team who has been and will continue to help lead this research as it develops over the next couple of years. Namely Tori Hanley, Randall Hughes, Teresa Davenport, and Chris Bailey. And as we'll discuss in our talk today, the phase of work we are launching with the support of the Lenfest program builds off of many years of work, and that work would not have been possible without a long list of collaborators, partners, and past researchers who've helped make this work possible. Some of those collaborators are listed here, but there's many, many others who have contributed through past research and or data collection that our work builds off of. So today I'm going to start by giving you some background into why we've put so much effort into pursuing this type of work, as well as the types of benefits we see evolving from it. And then Philine will provide an overview of our work to date in estimating juvenile fish enhancement values from coastal habitats. And then finally, John's will get into the new phase of the work supported by Lenfest, where we're looking to refine these estimates by incorporating several biotic and environmental variables. So, as I'm sure most of you know, coastal marine habitats are among some of the most valuable on Earth, providing a diverse array and disproportionately high level of ecosystem services, those such as providing habitat for nursery and foraging fish, sequestering carbon, stabilizing shorelines, and removing excess nitrogen. Coastal habitats like wetlands are multiple value systems, and that they don't just do one thing, but they perform many of these processes simultaneously, and therefore provide a suite of values to humans. Unfortunately, many of these services are at risk as coastal habitats are threatened through impacts like overfishing, habitat loss, and pollution. And salt marshes, seagrasses, and oysters are no exception to that. All of them have experienced significant losses in the U.S. And globally. These impacts to the habitats affect the benefits they provide to humans, putting people and communities at risk of significant economic and social loss. Fortunately, the impact and loss of these critical habitats has not been ignored or overlooked. Restoring and conserving the health of coastal habitats is now a preeminent global and environmental and societal priority. In the U.S. alone, hundreds of millions of public and private dollars have been invested to restore and protect coastal marine habitats, and it's the role of several federal, state, and local marine resource management agencies to consider these habitats when we're managing these resources. These resource management decisions and the public and private investments made towards these habitats are meant to recover and or conserve the ecosystem services the habitats provide. And while a large and growing array of science continues to confirm that these habitats are critically important and exceedingly valuable, this science is actually rarely translated into the language that helps drive change in the way we evaluate and manage for our many needs. And we feel that efforts to integrate ecosystem service benefits into decision-making require a more detailed, targeted approach, focusing on drivers for sustainable use, protection, and restoration of the ecosystems. This approach to describe in quantitative terms and representing the spatial variability in the services these habitats do for us has been coined mapping ocean wealth by the Nature Conservancy. The idea behind mapping ocean wealth is allow us to make smarter investments and decisions concerning the marine environment by allowing us to account for their value and the distribution of those values across an array of spatial scales. And so central to this approach requires locally accurate, spatially explicit quantification of those ecosystem services using metrics that can be understood and utilized by decision makers at different scales and in different settings. John, Philine, myself, and many others have been working to do that. That is, to advance both the science and the applicability of these types of estimates for nearly a decade, and the history of the work goes back nearly 2 decades at this point. And like I said, we're really excited to be moving into this new phase of the work with Lenfest. We have 3 main objectives with this phase of the project. As Philine will describe next, one of our objectives is to increase the spatial extent of our current estimates of fish enhancement into the northeastern part of the U.S., so from about the mid-Atlantic region up to through Maine, where our current analysis is lacking. Another objective, as John's going to then describe, is to identify what biotic and abiotic factors have the greatest influence on these enhancement values across all of our ecoregions. And finally, as with each of our phases of work before this, we're always striving to advance the ways in which the work gets best applied to resource management and decision making. And 2 of the primary ways we're doing that is, firstly, we're currently building a decision support tool that allows users to quickly and efficiently access our fish enhancement values. And Philine's going to provide a little more information on those efforts. And secondly, as part of the Lenfest project, we've developed an advisory panel. The advisory panel consists of restoration, conservation and fisheries professionals from various agencies and organizations. And the role of the advisory panel is to just help us guide how this science can best be integrated into various research decision-making frameworks. And John will explain a little bit more about the advisory panel group. So with that, I'm going to pass the mic over to Lena, and I'll keep controlling the slides. So Lena, you can just tell me next when you're ready.
Great, thanks so much, Brian. And thank you everyone for joining us today. As Brian said, I'm going to briefly introduce you to the background work that we've been undertaking for the last few years together with NOAA, but which will be coming to a conclusion over the next few months, which was to estimate an area-based fish and mobile invertebrate productivity enhancement from seagrass, salt marsh, and oyster reefs in the northern Gulf of Mexico and on the Atlantic coast of the United States. And when we talk about productivity in this case, we're talking about productivity enhancement. So it's the amount of fish and mobile invertebrates like crabs that we get from these structured habitats over and above what we would expect to get if those habitats didn't exist. And as Brian also alluded to, this work has not just been the effort of this small team, but we've been really lucky to draw on a wide range of collaborators from academia. They're mainly listed here. But we're also really grateful to all the field ecologists and the fishery scientists who work in the States that have gone out and undertaken many, many hours of field sampling and then made those data available to us. And without those data, it wouldn't be possible to put them all together into this meta-analysis approach that teaches us so much. So we're really grateful to all of those as well, even though they remain unnamed here. Next. So the rationale behind this productivity enhancement model that we have been developing is that for species that really rely on these habitats as juveniles and recruit to these habitats in a greater abundance than to unstructured controls, where those habitats are limiting, if you were to add or preserve those habitats, you will be adding juveniles into the system. And if we look at this in a purely theoretical sense, what we're talking about is working within this red box on the left side of this theoretical curve. So essentially, we know that the fish that rely on these habitats, if we increase the extent of the habitat, we get more fish, so greater fish production. And at some point, theoretically, other things will actually be bottlenecking that species, that they wouldn't be able to continue to have more and more just because we add more and more habitat. There'll be other limiting factors. But in the habitats that we're looking at, in the regions that we're looking at, we're actually very confident that because these habitats are at such a reduced extent compared to historic, that we are functioning within this box for the species that are really reliant on these habitats. But it is a caveat that's worth bearing in mind, because if we were really successful at restoring these habitats, we couldn't continue to apply this model in perpetuity. Next. So the basis of all this work has been a really deep data dive. It's taken us a lot of years to get to where we are, and we're really excited about the data set we finally pulled together. There are a lot of data out there, but actually relatively few that have made it through our quite stringent data requirements. Because we're interested in the enhancement, the amount that we're getting over and above what you would get in the absence of this habitat, we rely on having paired samples. So a sample from our habitat of interest as well as from a nearby unstructured control. Because we're interested in having a per unit area estimate, we can only use sampling that gives us densities, so we know what area are samples and what abundance of fish or crabs they caught. And finally, because we're interested only in recruitment, we also have to be very confident that the data we're inputting represent those young of year individuals that are really benefiting from having this habitat present, and that wouldn't be present in the system without those habitats. And to achieve this, we've done a really comprehensive literature review, but we've also been fortunate enough to have communicated with some fantastic state-based fisheries managers that have shared their fisheries independent monitoring, and without them increasing our sample size, we wouldn't have as great a model as we really are getting out at this stage. Next. So to give you some insight into what our data looks like geographically, in the northern Gulf of Mexico, we have data for salt marsh, seagrass, and oyster reefs. I'm just showing one example here, but for our salt marsh data, it's got a very good spread across the whole of the northern Gulf of Mexico, where you have 17 data sets we've pulled data from, giving us several hundred data points. 53 species have been identified from some of these paired samples. And the sampling techniques that are used are really good at picking up these juvenile that we're interested in, these drop and throw traps. Next. So if we look at the Atlantic coast, we have data for oyster reefs from the whole Atlantic coast and for seagrasses. In the case of seagrasses, we have enough data that we've been able to split them into these two regions, the Mid-Atlantic and the South Atlantic region. In both of these cases, there's actually over 2,000 data points representing these paired seagrass control samples across their range, which has allowed us to identify over 100 species in each case. Not all of these are enhanced, it must be said. This is before we apply our rules-based approach. But we're getting a really good coverage of the biodiversity that is available in these habitats and in these systems. But you'll notice that this map doesn't extend north of Cape Cod. Next. This is the data area that we're really excited to be having the opportunity with Lenfest to explore a little bit further. Our initial data dive didn't uncover a lot, but we're very confident, having started again now with Lenfest looking in this area, that we are going to uncover more data that will meet our data requirements. And hopefully over the next couple of years, we'll be able to apply a similar model to this northeast region for seagrasses. Although there are quite a large number of data sets represented here, it's actually a relatively small number of total samples, so we are still digging in this area, but we know there's a lot of opportunities here and a lot of people examining seagrasses, so we're very confident and excited to have this opportunity to spend more time making sure we can do this. Next. So how we get the production from this massive database of densities is that we take the quantitative studies I've described and we standardize them to independent data points. So by this I mean we have them each represent a similar spatial and temporal unit. So if someone is going out and sampling 2 days in a row, the probability that those data are not truly independent because the same school of fish might still be swimming around. So what we have to do is accidentally aggregate them into independent temporal units such as seasons, and also make sure that we have enough spatial distance between our sampling locations that they're not influencing each other. So a lot of effort goes in after we've got these data to standardize them so that they're all contributing meaningfully to the model and not swamping them. We take these data sets and we look across all of the data to identify species that are consistently found at greater abundances on our structured habitat as opposed to off. And we take those species that we then see as being enhanced and fit a long normal distribution using Bayesian methods to those raw data. And essentially what we mean by that is we ask the model to tell us what the shape of the distribution of probabilities of finding a species at a specific density is. So if we look at the graph on the right, on the y-axis we have a probability of finding a species, this species, at a given density, and the density is on the x-axis. So we let the data inform us about what this distribution looks like on a species-specific level. We then have the model randomly draw a seed or a starting density from this model distribution to give us a distribution of densities. And we then apply published growth rate and age-specific survival from fisheries models to each species in turn and to each model density of each species in turn. And that then gives us a species-specific production enhancement and an uncertainty based on that sampling around each of these values for each species. Next. What this looks like in reality is that for each species, we can then report out the number of juvenile, so young of year individuals that we find in the habitat per unit area over and above what we would find if that habitat was not present. So in this case, this is an example from the Gulf of Mexico oyster reefs. We find around 1.3 more blue crab juveniles in oyster reef as opposed to off. And across the lifetime of that crab, that will be that meter squared of habitat, it will be producing around 144 grams per meter squared per year. The graph above gives you a sense of how we can essentially accumulate that across a community. So for each species, we know how that habitat contributes productivity each year. If we look at the right-hand side of the curve, that's the amount we would get approximately from each meter squared of habitat on an annual basis for a habitat that has been existing for a long time. So if we conserve habitats, if we're interested in restoring habitats, it's obvious that we have to apply only the productivity that can be attributed to that specific patch of habitat. So in year one, we can only apply the productivity from those juveniles that have recruited in year one. Whereas in year two, we can apply the productivity that is produced from the year two recruitment, as well as the growth from the individuals that are still alive in the system as a result of the habitat being there last year. And that's why we see this curve that builds up biomass over time. So this is a question of whether or not you want to apply it to restoration or preservation of habitats. But it's a useful insight into how we're building these models to get to these annual numbers. Next. So as Bryan mentioned, really why we're in this isn't just to understand the ecology of these systems and to come up with estimates, but to put them in the hands of users so that they can inform decision making. And so as part of this previous work with NOAA, TNC have been developing a web-based tool to allow people to explore what our models mean on a spatial level for them. So I'm just going to spend the next couple of minutes walking you through what this tool looks like and what the outputs can be. So this is the front page of our tool. You'll see that there's a map of the area of the northern Gulf of Mexico and the Atlantic coast. And next. You can zoom in on this map. In this case, we're going to be zooming into Pensacola Bay. Next. And you'll see that the green represents the mapped area of seagrass in this area, and the pink represents salt marsh edge. And the reason it represents salt marsh edge and not salt marsh is that the salt marsh fish data that we have represents salt marsh within 3 meters of the edge or 1 meter outside of the salt marsh. So essentially the area of salt marsh that fish really interact with because salt marsh tends to drain completely at high tide. So that's why it's the edge that's mapped here and not the full salt marsh area. In this tool, you can then select, as you'll see on the left, whether or not you are interested in understanding the productivity from salt marsh edge or from seagrass. You can also decide whether or not you would like your metrics to be English or your units to be English or metric. And you can decide whether or not you would like to click on a study area onto the map, so use your clicker to actually draw a polygon, or whether you would like to directly enter an area of that habitat that you would like to, for example, restore or protect, if you know what that area is. So in this case, I click on Draw Study Area. Next. And you'll see here that draws a polygon using separate clickers. And then the tool works out for me not only the area of my polygon, but more importantly, the area of seagrass within that polygon. In this case, I've opted to look at seagrass. Next. The next stage is to select which fish species are present. And the reason I can do this is because the models that we feed in are for the entire region, but we wouldn't expect every fish that is enhanced by seagrass across the whole of the northern Gulf of Mexico to be present at every site in the northern Gulf of Mexico. So this allows the user to select the species that they know are present at their site, so that they're not falsely attributing value to species that aren't present. You'll also note these aren't real species yet. These are sample data for testing the tool. Next. And the next thing you can do is then ask it to give you the results. And the results will report out the number of new recruits you have entering your system every year as a result of that habitat that you've highlighted, or the amount of production you get on an annual basis from that habitat being present. You can also then download these data so that you can share them with your friends or look at them side by side if you're looking at more than one area. Next. We got really great feedback from this tool when we tested it with various partners. And there's a lot of utility for this tool, but we also got some fantastic questions from the feedback. And some of those questions were things like, we don't always restore habitat from nothing. We often actually improve the quality of habitat rather than restoring from nothing. Is that something that your model can help us with? Or we have a range of salinities across our system, and we know the fish populations look slightly different in the different parts of the bay. Is that something that your model can help us with? And the answer to that question currently is no. Actually, our models are across the whole region, and they don't account for this variability yet. But that's why we're so excited to be working with Lenfest over the next couple of years to try and answer some of these questions and build them into a format, again, that managers can use so that they can apply them in this broader range of situations. And so with that, I'm going to hand over to John to explain a little bit more about how we're going to do that.
A fair number of studies that have begun to investigate how properties of a estuarine habitat might influence the fish communities that use it. And we also know that the environmental factors can be important. So we're at a very nice pivotal point here. We assembled a lot of information about how fish use these habitats over the past really 2 decades, but we're now ready to kind of put it in a more realistic context, so to speak, so we can think about, how properties of a seagrass bed or an oyster reef or salt marsh might influence how it functions as habitat, as well as how important environmental variables might dictate the types of communities or organisms that use these habitats. And so just to kind of slowly introduce you to some of the habitats or some of the environmental factors that we're interested in, Bryan, if you would go to the next slide. There's many factors that we're going to be considering, but just to name a few, certainly salinity can play an important role in the types of organisms that utilize each of these different habitats. And we know that all of them can range across a pretty wide gradient in salinity. So we certainly want to think about where kind of in that estuary a patch of habitat is located, so we can think more about the types of fishes that might use it. Next. Another important variable is water depth, and this is particularly important with oyster reefs. We know that restoration is often conducted at, all the way from the intertidal to the, down several meters, and this can influence the types of communities that might utilize it, as well as the physical conditions that a reef might experience, such as bottom water hypoxia, that might influence how organisms use that reef. Next. Next, Bryan.
Should be on the intertidal versus subtidal slide. That's where I'm at.
Not seeing that.
Bryan, maybe try going back. I'm seeing intertidal versus subtidal on this end. If you want to maybe go back one slide and then go forward again and see if it resets.
It's working now. I see it now. Thank you, Bryan. So so here you see intertidal versus subtidal oyster reefs. This is another important variable. That we have oyster reefs in both the intertidal and subtidal, but often varying geographically where we find one or the other. This is an important factor for understanding how what we might expect a piece of habitat to produce in terms of augmented fish production. All right, go ahead to the next, please. So here we summarize the environmental factors that we're going to consider, and you can see that for many of them, we're going to consider them across the suite of habitats, but then for some, such as subtidal versus intertidal, it's really only specific to oyster reefs. All right, next. So in addition to those environmental factors, we want to consider a suite of habitat properties. We have information about where restoration's been conducted from a wide range of studies. And so we have the properties of the different habitats as well. So we can begin to think about how do things like oyster density or shoot density oyster or plant biomass or the percent cover of a habitat, how does that correspond with the amount of augmented density of a fish on a particular piece of habitat, so that we can then begin to explore, is there a relationship between any of these variables and augmented fish density, which would then translate to greater or less augmented fish production. All right, next. And we're also very interested in, where possible, comparing restored and natural habitats. Many studies have been conducted in both restored and natural habitats, which provides an opportunity to compare potentially how the fish communities might differ amongst these different types of habitats. Obviously, the goal of restoration is to recover the natural communities that exist in natural habitats, but it's challenging to really get at what potentially drives that. So I think this study really allows us, both with restored versus natural habitat comparisons, but also all of these other variables, to try to get at, potentially when are restored patches functioning similar to natural habitats, and when are they not, and potentially why. All right, next. And then obviously another important factor that Lena alluded to earlier in the talk is that habitat patch size, and particularly aerial extent, can have important roles in determining whether or not fish densities would be augmented. At the level of patch size, we might expect different species to use different size patches, but when we're talking about the entire ecosystem, obviously the aerial extent of a particular habitat can be important, because we expect habitat to be limiting at lower levels of aerial extent, but obviously other factors might come in and be important once we've actually reestablished habitat within a system. All right, next. So here you see a summary of the habitat properties that we're going to consider, and for things like density and percent cover and height and biomass, we're going to be exploring each of those, but obviously we're going to look for the best proxy for these four different highly covariating factors. And then with restored and natural habitat and aerial extent, this really gives us a better handle on how properties in general of a habitat might affect the types of fish communities that we see, and the degree to which they augment fish production. All right, next. So as a last word, Bryan alluded to the fact early on that our goal with this project is not just to focus in on the research, but it's also to try to figure out how we can push it forward into the decision-making and habitat management arenas. And we've assembled a great advisory panel that spans a wide range of fisheries and habitat management arenas to help us apply this work. I think with that, we're ready to take questions.
Great. All right. Well,
I'll pass the controls to you as well.
Great. Sounds great, Bryan. Thank you. And thank you to you, Lena and John, for a great presentation, and for certainly bringing us up to speed on this exciting work. So let me share my screen here quickly. Okay, and I think, yep, so again, if folks have questions, feel free to submit them via the Q&A panel on WebEx on the right-hand side of your screen, and we will get through as many as we can here. So I guess we can just get right to it and get started. The first question is more of a definitions question, and it's simply, what is meant by enhanced species?
Lena, you want to answer that?
Hello. Yeah, I'll take that. Sorry, I was muted. By enhanced species, we specifically mean species that are found more commonly than not at a higher abundance on the structured habitat as opposed to off, that have a mean positive density enhanced relative to the unstructured control, and for which there is evidence that they biologically would be benefiting from structured habitat at an early juvenile life history stage. So we're really confident that these species are actually benefiting from that structured habitat when they're at that recruitment phase into the system.
Great, thanks, Lena. So before I go to the next question, I just want to make a note. If you are submitting questions via the Q&A, please write them publicly as opposed to privately to me, just because my computer is currently presenting the closing slide here. So just to make sure we see your question, please submit them so everybody can see them. Our next question is a bit more technical. So in your secondary productivity estimates, how do you account for the uncertainty, error, or variability inherent to the calculations or assumptions, such as spatial or temporal variability in recruitment and growth rates? I can read that again if you need me to.
Go ahead, Lena. Willie, I think Lena's on mute.
Oh, okay. 1 second.
I've enjoyed talking to myself. Thanks. Sorry about that. When uncertainty, source of uncertainty. We essentially provide the information about the uncertainty. So when we model the distributions, we don't just include the means, we include the standard errors that around each of those individual means. So that informs our Bayesian probability distribution. And then essentially what the model does is by drawing numbers from that distribution, what it's taking into account is the error that's inherent in that underlying data is going to be represented in the width of the distribution. And we then put that forward into our productivity estimates, and we can then present those as standard errors, or once we've communicated with people and worked out exactly what is the best method of understanding that, we can put it potentially in other units as well. But essentially, we present what the uncertainty is, and something that we can work towards with Lenfest is breaking down exactly what those sources of variability are. That is something that we'd like to look into. But at this stage, those sources of uncertainty in the web-based tool are all lumped together, but there is the possibility of potentially disaggregating them at a later stage with some further research. That's something we're actively exploring.
Great, thanks, Lena, for that explanation. Our next question: why are you only considering subtidal seagrass?
I can answer that. So that is based on the majority of our studies thus far. If we were able to get enough fish use of intertidal seagrass habitat data, then we would certainly explore that as well.
Thanks for that clarification, John. Our next question: does the model track the enhanced productivity over time to see if there is a plateau or decline after a certain point? So maybe like a diminishing marginal return or something along those lines.
Lena, do you want to speak to that?
I'm in trouble unmuting. I'll try. I'll try, John, but you're very welcome to leap in. In a sense, the model doesn't do that now at the moment. What it does allow us to do is look at how that productivity accrues over time following restoration, from the point at which that habitat is actually functioning. And it allows us to account for interannual variability in terms of that will be incorporated in that error that I just discussed previously. But we don't currently think about plateauing over time. I'm not sure I completely understand if that's about when you increase habitats. As in that graph that I showed with the theoretical curve, we don't know the shape of that curve, and that's not something that we're currently able to explore because there isn't the data available to do so. So I don't know if one of my three answers answered your question. But John, if you understood anything else, feel free to jump in.
So I would just add to that Lena showed a graph where we showed each of the fish through time, and as we kind of move to the right, she shows like a steady state that a reef would reach, where we expect that once fish have recruited and fully matured, each new age class comes in. Right now, we're assuming that's kind of the max steady state that you would expect a reef to, or a seagrass or salt marsh to produce. There is the possibility to combine some of values that I was discussing, as if you were actively monitoring a piece of habitat and showed that changes occurred through time, you might actually be able to adjust the model. But I feel like that's pretty far down the road.
Great. Thanks, Lena and John. Our next question, have you considered or are you extending your productivity estimates to economic value?
I could answer that. That answer would be yes. We've done a little bit of economic modeling in the past, and certainly our goal here is to extend the work that we've done in oyster reefs to seagrass and salt marsh habitat, and eventually to be able to scale the economic values relative to environmental properties and habitat properties.
Great. Thanks, John. Bryan, I think this one is for you, and the question is the online tool for habitat area and species productivity available yet?
So we're currently testing it. We just got done doing a survey to a number of different stakeholder groups to get some feedback on the user interface part of it. And so we just got all that data back, and we're incorporating those changes now. And so we're planning on releasing it to the public. Hopefully, we're still picking on the date, but potentially towards the end of this calendar year is when we're expecting to have it launched.
Great, thanks, Bryan. Our next question, are there any species that show a preference for the unstructured habitats? And if so, do they count against the productivity calculations?
That's a good question.
Yeah, I can take that. So we can't quite do it in that way because the unstructured habitat isn't limiting. So we do find species that prefer the unstructured habitat in that they're found in a greater abundance than the unstructured habitat rather than the structured control. But that doesn't translate to
Sorry, Lena, just going to interrupt for 1 second. We have a little bit of feedback if somebody isn't muted. Okay, sound okay now.
I'll start again just in case. But essentially, yes, there are species that are found in greater abundance on the unstructured control as opposed to in the structured habitat. But our unstructured control doesn't meet the requirements of the model in that it's usually very abundant and not limiting. So it wouldn't be appropriate to apply the same approach to those species and say that if there was more seagrass, it would definitely be impacting those species that like unstructured habitats.
Great, thanks. Our next question. This is actually a similar question. Are you finding any species that do not have enhanced productivity in the salt marsh, oyster reef, or seagrass habitats? And do these habitats actually have a negative consequence for some species?
Yeah, I think the same answer probably applies. Yes, but I wouldn't assume that there was a negative impact on those species.
Got it. Thank you. Bryan, this is a bit of a follow-up on a previous question about the estimates. And the question is, when will the species and habitat-specific production estimates be available for a given location and region so that they might be applied and refined?
Yeah, so we have a version of the oyster estimates that are available now. And those can be found, through the published literature, of course, and Philine is the lead author on that. That was a 2016 paper, and those are also available in a more user-friendly format on our Mapping Ocean Wealth website. So if you Google search Mapping Ocean Wealth and then look for the resource tools, you can find those in there. And then, like I said, the salt marsh seagrass tools that we're working on now are currently under construction, and we're hoping to finish that by the end of this year. And then, as well as putting that data out in other formats, like the actual peer-reviewed publications that John, myself, and Lena and our other partners are currently working on as we speak. And those hopefully will be done by the end of the calendar year as well. And then, of course, the Lenfest variables, by the Lenfest variables, I mean, as we go deeper and start to understand some of those biotic and abiotic factors, we're not planning to have any of that analysis done for 2 to 3 years from now.
Great, thanks for that. Thanks for that clarification, Brian. That is currently all of the questions that we have in the queue. So if anyone does have any additional questions, now would be a great time to submit them. Just to note that on the slide, you do see that we've added the contact information for the lead PIs, as well as my information as well, if you have any questions about the project. So, we certainly look forward to being in touch with folks moving forward. But we'll give a few minutes here if anybody has any final questions before we wrap up. We have one question from folks here in the room at Lenfest, so I'm going to have him say it aloud here.
Hey there, I hope you can hear me down the other end of the room. Yeah, thanks so much for the super interesting work and congratulations. I'm wondering what you would say to somebody from a different part of the world or a different region, are there any elements of your results that you think could be transferable or useful to somebody in a different region? So clearly, I would imagine a lot of your quantitative results are specific to a particular place and species, but are there any sort of generalized results, like maybe, like certain types of species respond faster than others, or certain types of habitat more valuable than others, or just any sort of general findings that might be of interest to someone working somewhere else?
I can answer a little bit to that, in that that's not something, comparative habitats isn't something we've explored yet, but it is on the cards, because, it would be silly not to ask that question. But we don't have any results. Whether or not they're applicable across regions would be another question. But I would say that we have applied this approach to seagrasses in southern Australia and produced their own values for those. And so anywhere where it's possible to build off a large enough data set of paired, structured and unstructured habitat, and where your structured habitat or habitat of interest meets the requirement that it is threatened, then it would be possible to apply this approach and derive those values for the habitat. And like I say, we have applied that in other places. Those data don't exist worldwide, but anywhere that they do apply, it's quite straightforward to apply this methodology.
Great, thanks, Lena. We did have one more question come in for the piece that you presented, John. There's a question: what about proximity or closeness to other habitats, such as habitat redundancy, as another factor that could influence restoration effectiveness? So, perhaps, mutual benefits or things of that nature.
Yeah, that's definitely something that we're very interested in. We don't have a lot of data yet on that, but it is something where it's definitely in the back of my mind, because it dates back to my dissertation research. Is a question I was very interested in at a smaller scale, of course. But I would say that the degree to which we can use existing information around where habitats are spatially located, such as some of the tools the TNC has developed, we might be able to begin to get at that. I didn't mention it as an explicit variable because certainly we don't have enough information on that in the database that we've been assembling, because it just often is not available, unfortunately.
Great, thanks, John. All right, one more question has come in, which is there going to be any consideration of a habitat's geographic locations with respect to climate change and more rapid changes in certain places?
We haven't considered that explicitly, but because we're considering variables like temperature, and because there is a lot of information out there on where we expect climate change to occur more versus less rapidly, it's certainly something I think we could begin to think about after our analyses are complete.
Great, thanks, John. All right, one more has come in. do you see any differences in production enhancement between oyster and seagrass habitats? If so, do you have thoughts about how this might influence restoration decisions for a site, given that a plot cannot be both oyster reef and seagrass, for instance?
Yes, with our preliminary analysis, we do see differences between them. We see differences in total productivity, but interestingly also differences between species, so different communities. And so in that sense, I think when all the data are in hand, we can explore a little bit further what that means. Obviously, there might be situations where they could be viewed as competing, but given that we already know just from this preliminary analysis that although there is some redundancy, there are different species that are enhanced in each of those. I think it lends itself more to thinking about it as a landscape and a mosaic rather than necessarily in competition. And I think that's one way that frequently I feel restoration managers are thinking and moving towards as well. So hopefully that is something that supports our current understanding rather than being seen as something that's necessarily antagonistic. But we'll have to see how.
And just to add to that, just to add to that, given that these habitats provide a whole range of different services, I think you would be really remiss to just say, okay, we're going to go with this habitat because we think it produces the most fish.
Great, thanks, guys. All right, so that's all we have in the queue for now. We have about 8 minutes left, so I'm gonna leave things for a minute or two here and see if any final questions come in, and if not, we can close the webinar. All right, we're not seeing any more questions come in, so I think we can go ahead and close the webinar now. Again, thanks to Bryan, Lena, and John for the great presentation, and thanks to all of you for logging on today and attending the webinar and for the great questions. Again, we're really excited about this project to be getting underway, and hope that you continue to stay engaged with us. If you have any further questions about the project or about Lenfest in general, please feel free to reach out to me. My email address is on the bottom of the slide that you see right now. And of course, you can also feel free to reach out to the research team. So thank you all so much again, and we look forward to being in touch soon.